Hi, I am Sapir Margalit from Tel Aviv University, and I'll present our new study in which we used long-reads data to capture simultaneous methylation status in enhancers and promoters for the convolution of cell mixtures. DNA methylation is an epigenetic DNA modification that occurs in the denucleotide sequence CpG. Together with other epigenetic modifications, it is responsible for the variability in function and structure of the different cells in our body that all share the same sequence. Methylation in regulatory sequence elements is an important regulator of gene expression. It is known for many years that hypomethylated promoters are considered permissive for transcription initiation, while hypermethylation at promoters generally leads to gene expression suppression. But now we know that this is not the full picture. When a promoter is unmethylated across multiple cell types, enhancer methylation becomes the key regulator of gene expression and defines cell type-specific transcription levels. Enhancers are usually located away from their target promoters and reach them via 3D DNA looping. Enhancer methylation is also very relevant in disease states. Epigenome-wide association studies have shown that predisposition to common human disease, mainly cancer, is frequently associated with specific methylation signatures in distal enhancers. Here we can see the methylation level of a specific enhancer and the expression of the corresponding gene in normal and cancerous breast tissues. However, association studies are usually based on short-reads data from arrays or next-generation sequencing, which only assess enhancer promoter methylation on the population level, averaging out any differences between the individual cells in the studied sample. Long reads present a unique opportunity to study the coexistence of methylation in a promoter and its enhancers along the same DNA molecule, in effect providing single cell level information for the studied locus. Optical mapping is a long reads technology that currently provides the longest reads in the highest coverage. These reads are long enough to capture promoters and their distal enhancers simultaneously. With the Bionanogenomics optical mapping system, we use sequence-specific fluorescence labeling to create a genetic barcode and compare the barcode pattern on the single molecules to the theoretical barcode of the reference genome for alignment. We then turn to label the methylome. We use a specific enzymatic reaction that covers a reduced representation of the human methylome. Once the DNA molecules are duly labeled, they are stretched and linearized in nanochannels and are imaged as they translocate through them. This is how methylome maps look like. This is a region in chromosome 17 in the human genome. The blue wide strip with the orange lines on it is the reference genome with the barcode. The thin gray lines are the individual DNA molecules aligned to this region. The orange dots are barcode labels, and the dark dots indicate unmethylated CpG sites. This region contains a gene, TP53, and its promoter, and 3 of its enhancers. We can see that the promoter and 2 enhancers are very labeled, and that means that they are not methylated, and the third enhancer is methylated. Our goal in this project was to deconvolute cell mixtures based on the combinations of the methylation signals in linked enhancers and promoters. Such deconvolution can be of interest, for example, in cancer states, in which a population of cells can gradually acquire epigenetic transformations, and the detailed single-cell enhancer-promoter methylation profile may provide valuable information for studying the evolution of early carcinogenesis. Each enhancer-promoter pair can exhibit one of 4 possible methylation combinations. We count the number of molecules presenting each combination in different samples. These counts, or their respective proportions, can serve as signatures of the sample. In this project, we used synthetic binary mixtures. That means that molecules from two samples were randomly computationally mixed. These mixtures are referred to as a test set, while other molecules from the separate samples not taken to the test set were kept pure and served as training sets. The mixing was performed in several known ratios, and several deconvolution methods were suggested in order to find this ratio. The first method uses a probabilistic model in which each molecule in a mixed experiment is chosen from one of the samples, either A or B, based on the mixing ratio. Then the methylation status of the molecule is chosen. And given the sample of origin, we know the probabilities of the different methylation combinations from the training set. This problem is much like problem of dice in a basket, where the sample of origin is like the color, and the methylation combination is like the numbers on the dice. We want to estimate the mixing ratio by maximizing the likelihood function so that the observed data would be most probable. This is the log likelihood function of the problem, and since it is concave, we can find its maximum by using a simple gradient descent algorithm. A second method was to first select a resolution for deconvolution and assemble linear combinations of the two training sets with all possible mixing ratios according to this resolution. Then we want to find the mixing ratio that yields the minimal difference between the test set and linear combination of training sets. Two criteria were used for this minimization. The first is the sum over all enhancer promoter pairs and over all methylation combinations of the square difference between the test set and the linear combination of the training sets. The second is the KL divergence between the test set and the linear combination of the training sets. The third method is a local method that was calculated separately for each enhancer promoter pair. Each pair can be defined by three four-dimensional vectors, one for each of the pure training sets and one for the mixed test set. In order to find the mixing ratio, the difference between the test vector and one of the training vectors is projected on the difference between the two training vectors. The proportion of the size of the projection out of the size of the vector connecting the trains is the mixing ratio with respect to sample B. The final reported result would be the average over all enhancer promoter pairs. Two types of mixtures were evaluated and deconvolved. The first mixture was composed of two different cell types, B lymphocytes and myoblast cells. Over 500,000 single molecules comprised each test set. These molecules covered over 100,000 enhancer promoter pairs that were linked by the GEM method. The median length of molecules is over 230 kilobase pairs. The median distance between the enhancers and promoters in the covered pairs is over 50 kilobase pairs. Each pair in a test set was covered by 30 to 120 different molecules, with median coverage of around 50. On the top graph, you can see the calculated mixing ratios in the different methods as a function of the real mixing ratios in 11 different compositions tested. The bottom graph shows the average error in the mixing ratios in these measurements. The results of 3 out of the 4 methods are accurate. Maximum likelihood estimation provided the most accurate deconvolution with only 1% mean error, while vector projections provided the least accurate deconvolution. We continued to a second mixture, which is a more subtle scenario. It is composed of only myoblast cells, but from two different individuals. It's the same cell type, so most of the methylome is supposed to be similar. We can see here that, in this case, the results of deconvolution are not as accurate as before, with around 6% mean error. In order to improve these results, we reasoned that not all pairs contribute equally to the discrimination between sample types, and relying only on a subset of most differentiating pairs while filtering out the rest has potential to improve the convolution accuracy and reduce noise. For this end, we wanted to select the most contributing pairs in a supervised manner. We ranked all the pairs in the dataset according to their ability to discriminate between the training sets, and then analyzed the mixtures again with varying subsets of best pairs. The ranking criteria are Euclidean distances, KL divergence, or coverage-weighted KL divergence between the two training sets. Coverage-weighted KL divergence assumes that higher coverage provides more reliable information. These are the results after the selection of best pairs. The y-axis is the mean error over 11 different compositions, and the x-axis is the log 10 of the number of highest ranking pairs used. We can see here several combinations of the convolution method and ranking method. In the first mixture of two different cell types, we can see that the more pairs we add, the more accurate the deconvolution becomes. And that is reasonable since their methylomes are very different. But in the second mixture, we can see that there is an optimum at around 100 pairs. And above it, only noise is added. So this selection allowed us to decrease the error in the two myoblasts mixture to only 1.4%, which is low considering the coverage of the experiment. To summarize, long-read methods are emerging technologies that yield new types of data. Single-molecule methylome maps is one of them. Methylation signatures in linked enhancer-promoter pairs can be used for accurate deconvolution of a binary mixture. The best method for deconvolution was shown to be maximum likelihood estimation. For samples with similar methylomes, supervised selection of best pairs can improve deconvolution accuracy. This method is not limited to bionanogenomics optical maps only, but can also be applied to other long-read data. For example, applying it to Oxford nanopore sequencing data would be interesting to pursue in the future. Thank you very much for listening, and you can read more about this study in the new Proceedings issue of Bioinformatics.